My name is Rebecca Ihrie. I'm an assistant professor of cell and developmental biology with a secondary appointment in neurological surgery. My lab is interested in understanding the stem cells of the pediatric and adult brain and their relationship to brain tumors. My lab really likes to think of ourselves as cytometrists. So we measure things on cells, and we have two approaches that we primarily use to do that. And we apply them across research model organisms. So we work a little bit on animal models of tumors. We work on stem cell derived models that are prepared by my colleagues. And we work directly on tissue that comes from the operating room. And we do two different things with these samples. One is to do imaging cytometry. So we do lots of microscopy-based analyses where we take thin slices of tissue sections or preparations of cells, stain them for proteins of interest. So we make them fluorescent for the things that we want to measure, and then we visualize them on a microscope. And this lets us see not just what the cells, whether they express the proteins of interest, but also where they are. So where are they within the brain tumor? Where are they with relation to other structures in the brain? And how might this be influencing their behavior? In addition to the imaging cytometry, we do a great deal of what's called flow cytometry. And here we still take millions of cells from individual samples or from individual patients, but instead of keeping them all within the intact tissue, we prepare them as a solution of millions of individual cells. So we separate them from each other. We label them for 35 to 40 different things that we're interested in simultaneously. And then we're able to measure how much of each of those things is expressed on each individual cell within the tumor. And what this really lets us do is try to understand the diversity of cellular responses that are present within tissues and tumors. And we're particularly interested in finding cells that, when there's a great deal of them within a tumor, that it correlates to especially good or especially bad outcome, with the idea that we can then understand that biology better, try to target the especially badly behaving cells, and try to understand the cells that are better responsive to therapy or to the immune system, and try to push the tumor in that direction. I started thinking about cancer biology during my undergraduate training because one of the courses that I took as a biochemistry major was inorganic chemistry, and the first inorganic compound I learned about was a chemotherapeutic, cisplatin, which is still being used today. And this, I had always been interested in science, but I really wanted to be able to harness science to attack human disease. So from inorganic chemistry, I became interested in cancer biology, which is what my PhD specialty is in. During my PhD, I studied some of the most mutated proteins in human cancer. And when I moved on to my postdoc and I knew I would be trying to start an independent lab, I wanted to think about which cancers were really in need of scientific intervention. Where had there not been change in therapies for a long time? And brain cancer was one of those cases. Brain tumors remain extremely difficult to treat. There have been relatively few changes to clinical standard of care. And at the time, especially about a decade ago, we understood very little about the biology of these tumors. So I saw that as a big area of need and decided to go specifically in that direction for my future research career. We are going in the right direction, and I think there are some exciting changes coming. Basic science has been critical to understanding, for example, where brain tumors even come from. For many decades, it was thought that there were no stem cells in the adult brain, and that we had no ability to make new neurons after damage or after brain injury. And we now realize that that is in fact incorrect. And all of those discoveries came from basic scientists carefully observing different model organisms. Funding is really critical to our research, and in particular, a lot of the cytometry techniques that we use very regularly now were only just starting to enter the field when my lab started at Vanderbilt. So one of the instruments that we use in a core facility here, when I came to Vanderbilt, it was the 15th such instrument in the world. And having the funding from private groups and from donors that are willing to take a risk on big ideas is critical to getting that type of project started, because you really have to build it from the ground up. You have to collect intraoperative specimens from patients that are willing to contribute tissue to research. You have to assemble everything and show that it's going to work before larger foundations are willing to take a chance. So private funding and individual donations and small foundations are critical to getting ideas started. And then the NIH and other large granting organizations are critical to making sure that that research, once you have a well-developed tool and a very clear hypothesis, can continue forward, that you can keep developing this over and over. Yes, so one of the key success stories that has come from my lab in the area of adult brain tumor research is identifying a feature of the tumor's location within the brain that we can see on MRI. So when the patient first comes in and has a preoperative MRI, we know something about if their tumor presents in a specific location, what are some of the possible factors that might be influencing outcome for that patient. So are they at risk for the tumor coming back sooner? Are they at risk for a more aggressive tumor? So this gives the surgeon some immediate information in terms of how aggressive they want to be in trying to resect that tumor. And it has also opened up a whole new area of biology in my lab. So now we're trying to understand, how do highly malignant tumor cells get to that location, and can we stop them from getting there? And once they arrive, does the environment around those cells make them more aggressive? On the pediatric brain tumor side, we've collaborated with clinicians here that treat a disorder called tuberous sclerosis complex to try to understand why some of these children are at risk for a much larger brain tumor than others. And in addition to understanding the core biology of the stem cell niche better because of this project, we think we've actually found a specific population of neural stem cells that is at risk to become this larger tumor. And what that means for patients is that eventually, through discovery efforts that are ongoing in the lab, we can try to find treatments that will selectively target those cells and spare the rest of the brain. So some of the things that I was really excited about when I was choosing where to start my lab were the close connection between the clinic and the basic sciences at Vanderbilt. I'm a basic scientist, and I love to think about growth factor pathways and the response of cells to native stimuli. But I also, as I said, wanted to connect that back to human disease. And to be able to do that in the brain, you have to work with neurologists and neurosurgeons if you want to understand what's going on in patients. So I was very excited about the close clinical connections with both the Children's Hospital and adult neurosurgery. I also was thrilled to come to somewhere with a really large, vibrant brain institute. So the cross-disciplinary neuroscience program at Vanderbilt was a particular strength that attracted me. I think one of the things that's most exciting for trainees when they realize it, and part of my job is to bring this across early on, is that once you enter the research lab, the science you are doing is the real thing. When you find something out, you might be the first person in the world to know that piece of information. And because of that, it behooves you to be very rigorous in testing every idea and thinking about it very critically and doing the best work you possibly can. But also that's a tremendously exciting thing, and you should embrace that and be excited about your results. And I think the other thing to remember is that just because something seems hard doesn't mean that it's not worth doing. Sometimes you are the person who needs to leap in and do that difficult thing. So I did my undergraduate degree in honors biochemistry at the University of Michigan, and I was really inspired both by advisors in the lab and an advisor whom I worked for teaching organic chemistry to younger students to move forward with an academic career. So I completed my degree at University of Michigan and then transitioned to cancer biology at Stanford University. And I was my graduate advisor's first graduate student. So I was really able to experience what is it like to build a lab from the ground up, because I was there from the first day onward. My postdoctoral research was at the University of California, San Francisco in the Department of Neurological Surgery and at the Regeneration Medicine Institute.